All right, so, you know, this is just an example of, and I won't go into too much detail, but a few years ago, Devendra and I found a new gene in a spelt wheat. We mapped it to chromosome 2A. That's what it looks like, the low phenotype, to 7 different pathotypes. You can see that it goes up and down a little bit, but they're all incompatible. We developed that's not true. It was it 65 or 71, Devendra? 60. There are actually 2 genes we found in this thing, so and I've made a mistake there, but it's actually LR65. The second one we called LR71. But we did exactly what I've told you. We took that spelt wheat, old gold or rot corn, we did multi-pathotype testing initially. We found something we knew was different. We crossed it by a susceptible. We ran it out. We actually went to an F3 population, I think. We did the phenotyping. We extracted the DNA. We ran markers. We mapped it to chromosome 2A. We then did a test of allelism, I think, with VPM, didn't we? So there was another gene described on 2A, LR37. So we did a test of allelism. We showed they were independent. And we went to Bob, and he gave us the designation LR65 for that gene. So this is the basis of what we do with characterising, mapping and finding new resistance genes. And if we're lucky to find a marker that's really, really close to the gene, we can use that marker to help select for the gene. This is what we call marker-assisted selection.
Another thing I would like to ask, the cost of these SNPs is, the number of SNPs you include in your experiment, it increases the cost as well. Would it not be wise to eliminate the short and long arm of chromosomes on the basis of few selected markers?
I'm not sure I follow your question, Javed.
I mean, if we have, for example, 2A chromosome,
yep.
For 2A we have got short arm and a long arm.
Mm-hmm.
So there would be specific markers for short arm and long arm.
There are. Yep.
Say if there are 100 SNPs on short arm and 200 on long arm. So instead of applying 300 markers, can't we go for, say, 2 or 3 markers for short arm and 2 and 3 distant ends, and then cut down the cost of our SNPs?
Yeah. Yeah, we can, and we call this selective genotyping. So instead of genotyping the entire population, there are tricks you can use that will cut down the cost by doing what we call selective genotyping. There are other tricks as well, and I'm not going to go into this, but there's another approach called bulk segregant analysis, where you actually make pools of DNA.
Yeah, I was just going to say my colleague Renee Prince from South Africa has done exactly that. She's got 105 SSR markers covering all the chromosome arms, and she calls it a low resolution genome scan. And she's been very successful in locating specific chromosomes or even arms just by doing a low-cost screen rather than starting out with hundreds.
Yeah.
So it's certainly a valid point.
Yeah. But to do that, we need to have the markers that are evenly distributed across the genome, and to get them we've had to develop these newer, better marker technologies. So we're now in a situation where we can do that, and it does reduce the cost a lot, and it's very powerful, and it's a lot quicker.
What exactly is the difference between this kind of genetic mapping, what they call fine mapping?
The high resolution mapping?
Yeah.
Yeah, so, well, really, the high resolution mapping is used for marker development, link marker development, and gene cloning. That's the really, I suppose, the holy grail is to actually get to the gene itself and then design a marker on the gene so it's 100% diagnostic or a perfect marker for the gene. To do that, we need to have what we call high resolution mapping. And typically for that, what we would do is initially, we have the gene, we would initially try to find 2 flanking markers within, say, a centimorgan each side, Devendra, I would think. So very, very closely, markers very, very closely flanking the gene. Then we would go to an F2 population of a resistant biosusceptible, and that F2 population would be at least 1,500 plants, possibly even more. And what we do, we don't even bother phenotyping the F2s quite often. We just extract the DNA and look for recombinants between those 2 markers. We know the gene's sitting in that region. We just look for recombinants in that region, and that becomes our high resolution mapping family. And out of that 1,000 or 2,000 plants, we might only have 10 or 15. We then phenotype the progeny of them, and we go in and look for a combination in the markers that lie between them to get closer and closer to the gene and hopefully end up landing on the gene. Is that the answer to your question?
Thank you.